Well, hello everyone. Welcome to this new webinar. So without further delay, let's get started. Let's start the webinar with the first lecture by Gerardo Mendizabal. Dr. Mendizabal holds a master's in computing and industrial mathematics and a PhD in computer science with over a decade of experience in artificial intelligence and computer vision. He is a research professor at the University of Guadalajara, where he directs the Laboratory of Autonomous Systems for Biotechnological Design, focusing on machine learning, deep learning, and biomedical innovation. In parallel, as Vice President of Product Engineering at Conceivable Life Sciences, he co-developed the first fully automated ICSI system, leading to a successful birth. His work bridges academia and industry and advancing AI-driven healthcare solutions. So Gerardo will deliver today a lecture titled From Manual to Automated, Designing the Next Generation IVF Labs, and he will explain what is the way to carry out this automation in the lab. So please, Dr. Mendizabal, the floor is yours.
Thank you very much for the invitation. It's a pleasure for me to be here today. I'm here to tell you a little bit about how we are designing the next generation IVF labs. But first, these are my affiliations. As I was presented, I'm VP of Exploration and Professor at the University of Guadalajara. So the success of an IVF treatment depends on many factors. For example, the age and health of the patients, the quality of the gametes, or the conditions of the lab. These factors set the stage, but they are not decisive. A determining factor is experience and the skills of the embryologists. Even with perfect conditions, if the procedure is not performing correctly, the treatment can fail. Therefore, embryologists require years of extensive training before they can safely and effectively perform IVF procedures. Their work demands not only technical mastery of delicate techniques, but also the ability to make critical judgments under pressure. Reaching this level of expertise involves rigorous education, countless hours of hands-on practice, and continuous refinement of the skills. Unfortunately, despite having the highly skilled embryologists, standardized protocols, and the state-of-the-art equipment, the outcomes can still fluctuate from one procedure to the next, even within the same clinic. A small difference in the techniques, timing, or individual judgment inevitably introduces inconsistency. This variability is a challenge not tied to a single clinic or practitioner. It's a universal reality in IVF. Moreover, creating an embryo through IVF is not a single procedure, but a chain of nearly 200 manual steps. From gamete preparation to fertilization and embryo culture, each step requires human intervention, precision, and judgment. This complexity not only makes the process highly demanding, but also increases the potential for variability and error. One of the most critical roles of the embryologist is the decision-making. At every stage of the IVF process, they are required to interpret observation, weigh options, and make choices that directly affect the outcome. From selecting the best sperm to inject or embryo to transfer, their decisions will carry a significant impact. In addition, the embryologists also rely on highly refined motor skills, preparing the cultural environment, transferring delicate cells from one dish to the other, performing integrate micro-manipulations or single-cell surgery. All these demands extraordinary steadiness and coordination. These tasks are carried out at the microscopic scale, looking through a microscope objectives. And here, even the smallest movement can determine success or failure. So we believe that automation and AI have the potential to make IVF more effective, consistent, and scalable. By reducing the reliance on manual step and subjective judgment, it can bring consistency where variability exists. precision where human limits appear, and scalability where the resources are constrained. In doing so, automation has the potential not only to improve outcomes, but also to transform how IVF is practiced worldwide. IVF, AI and automation have already entered the IVF market, and AI is primarily used to support embryologists and physicians in decision-making, while automation focuses on streamlined repetitive tasks. So let's talk about the first one, about AI being assistant. Thanks to the vast and growing volume of data generated in the IVF field, it has been possible to incorporate some of the most effective AI approaches, particularly those based on neural networks, and apply them to support data-driven decision-making. So now every microscope in the clinics have a digital camera, so that creates these huge databases where the images can be converted to numbers that can be understood by the computers, and these numbers create patterns, and these patterns is exactly what the computers can understand, in some cases even better than the humans. So these models can learn from these thousands of past cases, identifying these hidden patterns and outcomes that might not be visible. And this ultimately enhances the precision and consistency in the clinical decisions. Assistant systems support embryologists in decision-making by providing data-driven recommendations, such as embryo viability scores, sperm selection guidance, or alerts from environment monitoring. The important thing is they don't replace the human oversight. They enhance the decision-making. They help reduce the subjectivity and help with the standardization of the outcomes across the labs. There are already some companies that already offer some of these tools. Automation, on the other hand, has primarily been focused on smart culture systems and real-time monitoring of embryos through time-lapse imaging technologies. These systems enable continuous observation without disturbing the culture environment. This enhances consistency and decision-making. Another notable automation advancement is the cryo storage platform developed by Tomorrow Life Science, which ensures stable storage conditions and full traceability of gametes and embryos. This is aimed to significantly reduce the risk of error and improve the lab efficiency. While these systems have proven to be of value in the IVF lab, many routine labor-intensive tasks still depend on steady hands and stopwatch precision. For example, preparing culture dishes, exchanging media, denuding oocytes, performing embryo biopsies, loading big vitrification straws, thaw, warm, logging QC checks, and sharing specimens among incubators, microscopes, and cryo storage. Automating these steps will boost repetitivity, tighten standardization, and let the embryologists focus on higher-level clinical decisions. Fortunately, breakthroughs in ultra-precise robotic and cutting-edge optical systems now let us reimagine the IVF laboratory as an AI-guided, fully automated space, where micron-accurate robots steered by high-resolution imaging can handle even the most delicate embryology tasks with consistency data-driven precision. So this is exactly what we're doing in Conceivable Life Science. And over the past 2.5 years, we have developed some of this system based on artificial intelligence and robotics. One of the important aspects of what we're doing is that we are not trying to reinvent embryology. Instead, we are replicating the proven techniques that have already brought millions of babies into the world. However, now those are being executed by advanced robotics and AI that will deliver the same expertise with greater consistency and scale. Let me show you some examples of the first generation of systems that we developed during the past 2 years and that we used for our first pilot study. So in the left, you will see one system that is aimed to sperm preparation. You can see here a robotic pipette that is able to pick up the tips. It takes raw ejaculate and put it in one of these falcon tubes. The next step, it will dispose the tip and go take a new tip, and now it will take some media. And this is basically the swim out protocol that many of you are sure you are already familiar. So after some time, we take a sample of the supernatant and we put it on a new Falcon tube. So this is one way of doing the sperm preparation. We have another way that is based on custom microfluidic style, like similar to the CyMod. In this system, the robotic arm is capable of creating a specific pattern that will be used to do what we call the swim out. Here you can see this pattern. And the idea is to deposit the raw ejaculate in certain droplets, and then the sperms will swim through these channels to end up at a pilot droplet. And then we monitor these droplets using microscopy, and when we reach the concentration we need, then the system will cut the bridges and make sure that this concentration is being retained in the center droplet. So this is sperm preparation. We also have a system that we use for doing egg finding. Is the system that you see on the left. I'm going to show you one example of how the egg finding in follicular fluid works. Here, the system is scanning. The AI detects a COC, and then it will position the plate. It will bring the pipette, and then automatically the pipette will determine how to expose some media and then aspirate the COC, which is gonna be bring to a new clean handle media. So you can notice that every time that the system performs an action, there's this window that allows the user who is supervising to take control if he believes that is necessary. So this is a very important step, because we still need we still want somebody to make sure that everything's happening the correct way. And in some cases, if any small adjustment is necessary, that they can perform it. Regarding insemination, we also developed the system that you can see in the left, that is based on a microscope, inverted microscope. But here we added a lot of AI that drives the complete HC procedure in an autonomous way. So this is a short video. The first thing that happens is that we immobilize the sperm using seed and a lace pulse. Then the AI automatically positions the needle, absorbs the sperm, stabilizes the needle, then it goes to another droplet where there's the egg. It finds the egg, change magnification, center the egg, hold it, bring the needle, deposit the sperm, and exit. So all this happens totally automatic, again, with the supervision of somebody that can intervene if necessary. So we already did this in the pilot study. We already used all of the systems that I'm presenting you. We just published a paper regarding the first live birth of this system that I just showed you, which was supervised from many miles away. This was from New York. The embryologist was Jack Cohen in New York, and then the system was in Guadalajara. So he was supervising this procedure, and that's what we call the remote ICSI. So this is the first in the world that happened. So I invite you to check this publication if you're interested. And we have a longer video that explains all the steps if you're interested. So we're very proud to announce that we have 20 world first live births using automation, either individually, one of the system, or in combination of the systems. So we just finished that preliminary pilot study, and we are now working on the next generation of this system. Alejandro is going to tell you more about that. And well, that's it from my side. Thank you very much for the opportunity to present on this wonderful platform. Thanks.
Thank you. Thank you very much. Really, really nice presentation.